Let's talk about histones, shall we? All right, the histone is going to be found in the nucleus. It's a group of 8 proteins. It's actually two copies of this particular unit, so there's the same thing going on in the back. Within a histone, there are 4 kinds of proteins. They are histone 2A, histone 2B, histone 3, and histone 4. In some other kinds of an animal, you would find a histone 1, but in humans, we have 2 copies of histone 2, is what it looks like. And so, different evolutionary path, but we end up at pretty much the same place. And so, on the back, there's going to be another copy of 2B, 2A, 4, and 3, right back there. Now, the histone is going to act like a spool for thread, which means that the DNA, which is this gray cylinder, is going to wrap around the histones. And when the DNA is wrapped up around the histones, it's protected, so it's less likely to get damaged, but it is also inaccessible. The histones are blocking access to the genes that are in the DNA that's wrapped around them. So, in order to control which genes you can actually use, histones have a big role to play. They kind of determine what you are able to use from your genes. Yeah, just to put it, very basically. So that's the role that histones play, and they have a very important role in diseases like cancer, for example. But also just general roles in which proteins you can make or not make. Because if you can't express the gene, you cannot make the protein. That is the idea. So you've actually got all these genes in every single cell in the body, but every cell has all the genes it would take to make a neuron or to make a liver cell or to make a muscle cell, for example. But you don't get access to all of those. And part of how the access is controlled is that we shut down parts of the DNA with histones. Also, if you are female, genetic female with 2X chromosomes, one out of those 2X chromosomes is going to end up silenced. And this is called X chromosome silencing, as you might imagine. And it's very interesting. It also happens in female animals. This is the idea behind why you get particular color patterns in female cats, for example. Because some of the color genes are located on the X chromosome, and they've got 2 of them. And so when a cell divides, each new cell is going to, completely at random, decide to shut down one or the other of its X chromosomes. And if the X chromosomes are saying to make 2 different fur colors on a cat, then you'll shut one off or you'll turn the other one on, and you'll get a little patch of color just like that. That's how we get calico cats, and why they are usually female. There are a few cases where you get male calico colored cats, but that's when there's been some kind of rearrangement and one of these genes has moved to a different chromosome. So it can happen, but that is not the most common path. Yeah. So let's talk histones and histone modifications. So the histone is a protein, and the histone protein has a large number of lysine and arginine amino acids. So you can see all the little Ks, K, 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 and then the odd R that's in here. The lysines and arginines on the histone tails, so there's a sort of a globular body of the histone that the DNA is going to wrap around, and then the tails are where you put all the modifications. The tails are what is going to control whether or not the genes that are wrapped around the histone are going to be expressed or not. So these modifications can kind of loosen the histone's grip and allow it to slide along so that you can express the genes, or they can kind of tighten up the histone's grip so that there's really no way to get at the DNA that's underneath the histone. And in the human body, we will shut down and open up great swaths of DNA. We'll shut down whole groups of genes at the same time, or open them up at the same time. All right, so this is a useful map, and what we're going to be doing is you're going to be filling in something like a table on the test. So we've got 4 kinds of modification. We've got a methylation, we've got an acetylation, we've got a ubiquitination, and we'll get to the other one it's coming up. And these modifications can happen at different sites. And so one of the things you're going to have to do for each modification is you're going to have to tell me one site where we do get a particular kind of modification. For example, lysine 20 of histone 4 can get methylated. Now, we're not going to go whole hog. We're not going to learn all of the proteins that read these methyl groups and acetyl groups out. This one is specifically looking at, I think, mostly the methyl groups. But this information's out there, and this information is kind of the next step. It's also important, but I figure for the purpose of this, we're just going to talk about, histones. So the process that controls whether or not you can express a gene is called epigenetics. And so epigenetics can actually end up causing phenotypes. There's a neat case with a mouse where one particular kind of just regular black laboratory mouse will have a white tip on its tail, or rather a pink tip on the end of its tail. And this is able to be inherited. Epigenetic changes can be passed on from one generation to another, although they do tend to decrease over multiple generations. So you can pass it on pretty strongly the first time, a little bit less the second time, third time, and eventually they will go away. The idea is that in evolution, epigenetic changes happen first. So first we change how we're using genes, and then second, some kind of mutation happens in the genome that locks an epigenetic change that's already happened into place. And that's an interesting theory. That would potentially help explain how a complex process like evolution can happen. And if that turns out to be correct, or at least partly correct, then it's all down to histone modification, which is what we're talking about. So epigenetics, that's really the goal of today, but we're going to study that through histones. And there are certainly other ways to look at epigenetics. There is modification of the DNA or RNA itself. Some of this can get methylated, for example, and modified in other ways like that. And you can also have epigenetics at the level of storing up a large amount of either messenger RNA or protein for some particular process. And so if you've got a lot already stored up, then it doesn't matter if you make a lot, continuously, because it's already right there, and the cell is able to respond very quickly. So let's dive into epigenetics. Okay, here are the groups. So we've got a methyl group, which is just your regular CH3. We've got an acetyl group, and remember there's a carbon there. So we got a methyl group on the end, a carbon here, and you've got a double bonded oxygen off of it. Phosphate can be attached to the histone tails as well. And that's just PO4 with either 3 negative charges, or when it's hooked up to something, it's going to use up one of those negatives, so it'll just have 2 negatives left over. The other general type of modification, we're going to lump these together, is going to be attaching either ubiquitin or the small ubiquitin-like modifying protein, which is called SUMO. And there are others that look like this and work kind of like this. These are little proteins that, as I think you'll remember from our talk on HPV, can be attached onto other proteins. And so, you can get ubiquitin attached to a histone, and it will have different effects. So, depending on exactly which amino acid of which histone we modify, we will have a very specific effect. And so there are some general trends here. Some of these modifications tend to lead toward shutting down genes or opening up genes or marking damage to the DNA that's next to the histone. But there is specific detail here as well. So any particular site is going to have its own basically histone code. Together you can call all of this the histone code. It's like another genetic code, rather an epigenetic code, that is sort of expressed on top of the regular genetic code. So in addition to, AUG coding for methionine, like that, in a regular protein, you've also got the fact that if you can't loosen the histone to get at the AUG, then you can't make the messenger RNA, and there will be no expression. Or if you really loosen it up, then expression is going to be higher. So very diverse biological functions, just like it says there. And if you want to read a little more, this is kind of neat, this plantEpigenome.org. down there at the bottom. All right, so what are some of these histone modifications? Well, it's actually more complicated than just saying, yeah, we attach a particular group to a particular thing. So in the case of acetylation, have a look at this. So we're starting out with lysine, and you'll remember lysine is one of the basic amino acids. Basic means it's going to carry a positive charge at kind of normal intracellular pH. That's the idea. And so here's our lysine with all its little carbons, and there's our NH3 at the end, and it's got a positive charge, and yes, that's what lysine would normally look like under ordinary intracellular conditions. But when we modify it, look at that. We're putting the acetyl group right onto there, displacing two of those hydrogens and using up the positive charge. So not only are we changing the size of the molecule from a relatively small molecule to a much larger molecule, by 50% larger, we are also changing the charge of that molecule. And so changing both of these things, the size and the charge, can have a big effect on whether or not a protein is able to bind to it. For each of these modifications, there are a set of proteins that write the modification, there are a set of proteins that erase the modification, and there are a set of proteins that read out the modification. Now, usually there are only a few proteins that do the writing and erasing, and so we can just cover those fairly easily. The proteins that actually bind to the modification, there are many, many proteins, and they're going to be very specific. They're only going to bind to a particular modification, to a particular lysine on a particular histone, for example. Yeah, so those are very hard to generalize, and so I'm not going to have you learn any of those. But if you end up going on in this field, that's going to be something that you will eventually need to get a handle on. So acetyl groups change the charge and change the size. Methyl groups, on the other hand, do not change the charge. They just change the size. But the other neat thing about methyl groups is that you can get 1, 2, or 3 methyl groups attached to these lysines. So you can get regular lysine with a positive charge, and notice the positive charge persists all the way through. And you're basically just displacing these 3 hydrogens. You can take each of the 3 hydrogens off one at a time and add on a methyl group to that. And you will get proteins that are only able to bind trimethyllysine on a particular amino acid, for example, of a particular histone. So there's a lot of complexity here, and not all of this is fully worked out. This is very much an area of developing science, and so this is an area to watch. We are learning new things about histones and their modifications all the time. But yeah, acetyl groups are going to change the charge, erase the charge basically, and methyl groups are going to leave the charge in place. I think that's important. So histones are modified by groups of other proteins, and so the things that add or subtract these molecules are going to have different names, but they're actually fairly easy to figure out, I would say. So an enzyme that adds an acetyl group is going to end in ASE, so this is ases because we're talking about a bunch of them, but an acetylase, or better yet, acetyltransferase, is going to transfer an acetyl group onto a protein, like this one, like a histone protein tail. And so there will be different acetyltransferases, but we're going to group them together and call them histone acetyltransferases, which is HAT for short, histone acetyltransferases. And when we're talking about erasing those acetyl groups, that's going to have to be a histone deacetylase. So that is HDAC, histone deacetylase for short. And then there are a lot of different proteins that can read these out, but there are so many of them that we are not going to cover any specific ones here. Just know that there are a lot. So for adding the methyl groups, we're going to have methyl transferases to add them, and they're going to be histone methyltransferases, or HMTs. For removing the methyl groups, we're going to have histone demethylases, so HDMs. And then for adding the phosphates, we're going to have, we normally would not call them a phosphorylase. Normally, We already have a very good word for a protein that adds a phosphate to another protein. It starts with a K. Do you remember what I'm talking about? Kinase, right? Yeah. So, kinases are the more common name for a protein that's going to add a phosphate group to a histone. And the ones that remove the phosphate group are generally going to be called phosphatases. So that's pretty normal. All right. So yeah, but there are writers, erasers, and readers that are adding, subtracting, and interpreting the different modifications to the histones and making the necessary changes to the way that the DNA under the histone or around the histone is going to be accessed. Okay, so here is a nice list of these modifying enzymes. Now, I think what you're going to want to do is build a table of some sort. This is part of the table, and we're going to need a little bit more information on there. So we need to know which amino acid is going to be modified, yes. We need to know what kind of modification we're talking about. We need to know what enzyme adds the modification, what enzyme takes away the modification, and we need to know a specific example of one of those. If it's a lysine, I need to know that lysine, a particular lysine on a particular histone is going to be modified. And then I need to know what is the effect when we modify that particular lysine on that particular histone. So amino acid, modification, writing, erasing, specific example, and effect. Those, I think, 6 parts. That's what the exam question is going to show. All right, so for lysine residues, you can get both acetylation and deacetylation with the histone acetyltransferases and histone deacetylases. For lysines, you can also methylate or demethylate them. So there are histone methyltransferases and histone demethylases. For lysine as well, you can add ubiquitin, and you can ubiquitinate or deubiquitinate. That's what we call the process of adding or subtracting a ubiquitin molecule from these. And so the enzyme that adds ubiquitin is called a ubiquitin ligase. Ligating means to stick something onto something else in molecular biology. For the removing enzyme, it's going to be a ubiquitin protease or a deubiquitinase. That's an acceptable alternative name. And then for adding phosphates. So phosphates are the ones that are not added to lysine. They have to be added to a group that has an OH on the end. And 2 of those are going to be serine and threonine. Lysine does not have one of those. It has a free amine on the end of it. And so the serine and threonine, and then you can modify either one of these. They look very similar. The threonine is just a little bit, it's 1 carbon longer than the serine. But the modification is going to be carried out. We're going to add a phosphate with an enzyme called a kinase, and we're going to take away a phosphate with an enzyme called a phosphatase. And lastly, arginine can also be modified. And so arginine is an enzyme that is, or rather an amino acid that's able to be methylated or demethylated only. So it cannot be acetylated. And the methylation is going to happen with a protein, arginine methyltransferase, so PRMT. And it's going to be demethylated with a, you can call it a histone demethylase. I think that's fine. Deaminase is a different form of the word, but I think demethylase, put the E back right up there where it belongs. I think that one's going to be easier to remember. So those are the enzymes that you're going to have to remember for the different modifications. You can add SUMO, which is the ubiquitin-like protein, small ubiquitin-like modifying protein. That's also added to a lysine, but we're going to forget about SUMO for the purpose of this. And you can add ADP ribose, which can only be attached to glutamate residues. ADP ribose is actually 2 ADP molecules, adenosine diphosphate, that are hooked up 5' to 5', so like front end to front end, which is weird because they normally hook front to back, like you join onto the back of the one in front. Just like train cars, just like, a conga line, just like anything sensible, really. But these end up sort of face to face, these ADP ribose molecules. And they are attached to proteins, and they do have big effects. And you will see ADP ribose addition and subtraction proteins heavily involved in processes such as cancer. Okay, so what are some of the roles for these? Well, you've got some modifications that are going to be involved in repression of genes, turning off certain genes. So that even if you make the right transcription factor, which should bind to the gene, it's not going to be able to bind because the histone is already stuck to that gene, and the histone's not moving. You're going to have activation. And so an example here would be that histone acetyltransferases are generally going to be activators. They are going to make it easier to get to the gene that's underneath. So think of the acetyl group as like loosening up the histone, so you can just slide it along and get to the genes that are underneath it. Then over here in the repressors, you're going to have the histone deacetylases. So acetyl groups generally are going to lead to more expression. Deacetylases are generally going to lead to less expression. But there are other ways that these modifications can be useful. For example, if you get a double-stranded DNA break, so this is the DNA damage, and we're assuming that this is caused by UV light or something. That's what the lightning bolt is meant to signify. The H2A point X, so is a particular version of an H2A, and this is one where we can get phosphorylation at the H2A point X. And phosphorylation there will actually mark this as the site where there is something wrong. So there's an H2A gene that I believe comes from the X chromosome. I think that's what the H2A.X means. And usually you use the normal version of the H2A, but every couple of these histones, you will have the H2A.X instead. And so that's what's in there. But for the purpose of describing what a histone is actually made of, I think an H2A is enough. But yeah, H2A.X phosphorylation is a signal that tells the cell that we need to bring in the repair enzymes to try to fix a bit of damage. So looking forward to the CRISPR-Cas9 lecture that's going to be coming up, CRISPR and Cas9 are going to be a way that we will be able to deliberately break double-stranded DNA. And once it breaks, then we're going to have H2A.X phosphorylation to mark that break, and that's going to start the repair process. And so we'll learn all about that when we get to CRISPR, but yeah, just this is a, not a call back, but like a call forward? I don't know, foreshadowing? Who knows what that is? All right. Okay, so acetylation. What does it do? Generally, acetylation is going to open up chromatin. Remember, acetylation is going to change the charge of the molecule. You're only going to be able to acetylate a lysine. And so, yeah, for example, here's a nice little example. So if you have histone 3, H3, lysine 27, the 27th lysine along, and you put an acetyl group on it, then that is going to activate the DNA in that area. And you would typically acetylate several histones in an area that you want to open up. At the same time, histone 3, lysine 27 can also be methylated. And if you put 3 methyl groups onto that thing, then that is going to totally inactivate that bit of chromatin. Chromatin means DNA in this case. So yeah, and it's going to tend to recruit proteins with bromodomains. That's a particular kind of domain. We don't need to know that for the purpose of the test, but it is actually true, and so yeah, that's useful. All right, so yeah. And remember that acetylation is going to be added by an acetyltransferase, and acetylation is going to be removed by histone deacetylase. So histone acetyltransferase, HAT, or histone deacetylase, HDAC. And remember the acronym, but try and write it out. That's better. All right. So There are multiple, superfamilies of histone deacetylases, which are going to shut down certain genes. So histone acetyltransferases open up genes, histone deacetylases shut down genes. A lot of these are going to be used in, for example, as cancer drugs. But some of these are actually, sold as beauty treatments. So you can actually change the way some of your histones deacetylate. Uh, or your histones are deacetylated and therefore shut down certain gene expression programs. That's pretty serious medicine. That's a little more serious than your average beauty treatment. And this is actually almost genetic medicine. Yeah, so use with caution, I would say. This would be something that I think you cannot get without a prescription. At least I would hope you cannot get without a prescription. All right, so that was acetylation. Acetylation is generally going to open up the chromatin, and then on the other side, we're going to have methylation. And now remember that the methylation does not change the charge of the lysine. It's still going to retain its positive charge, but the methylation is going to be on histone tails, and there can be 1, 2, or 3 methylation events there. So histone lysine methyltransferases are HKMTs. Histone arginine methyltransferases would be HRMTs. That just makes sense, I think. And there are going to be different effects depending on whether you have 1, 2, or 3 methyl groups on there. So there's a lot of subtlety in this part of the code. The methylation is also involved in this process in particular, which is X chromosome inactivation. And I think that is also kind of interesting. And we can cover that. All right, so let's start out with histone lysine methyltransferases, so really HKMTs. The K being the single letter amino acid code for lysine that you learned so long ago and are getting so much use out of. So I'm hoping you realize that that's paid off. Yeah. All right. Each of these histone lysine methyltransferases is very specific. So they're going to hit a certain lysine on a certain histone and no other lysines on any other histones in general. And some of these can put on just the first, second, or third of these things. Others are, I believe, a little more promiscuous, and they can add multiple methyl groups. And many of these contain a kind of methyltransferase domain, so that's very good. The readout is also going to be very specific. So here's another couple of examples. Histone 3. Lysine 4 with a single methyl group is going to have a different result from histone 3 and lysine 4 with 3 methyl groups in there. You'll be able to binds different proteins to each one of these depending on whether you have 1, 2, or 3 methyl groups. Here's the whole story on methylation, and I would say let's look for general trends and then pick out your very most favorite example, and then I think that's going to be all right. Okay, so over on this side, we're going to have inactivating modifications. So in general, let's see. If we want to inactivate an X chromosome, so that's going to be histone 3, lysine 9 methylation. That is going to, shut down the one of the X chromosomes, and the 2 X chromosomes actually line up, and we don't really know how they decide, but they apparently get to decide, and whichever one wins, the other one, I guess, gets methylated and shut down for the most part. Although a few little parts of that inactivated X chromosome will remain open. It's not completely transcriptionally silent. We do express a little bit from there. All right. So, let's see. Up here at the top, we've got histone 4, lysine 20 methyl group. That is going to shut down just an entire area of chromatin. And the inactive X chromosome is also going to have histone 3, lysine 29. I think this one's easy to read, to remember. Histone 3, K9. Think of a K9 as like a dog, right? Yeah, it's stupid, but why not? And so imagine you send your dog out to scare off all the replication machinery, and protect protect the DNA so it doesn't get expressed. Maybe that makes sense. Maybe it doesn't. I don't know. But yeah, in general. Now, other kinds of, for example, histone 3 and histone 4 arginine methylation are going to lead to, generally, activation of the chromatin that's underneath. So that is kind of the opposite path. But remember, this is arginine methylation leads to activation, and lysine methylation leads to shutting down the chromosomes. Whether we're adding methyl groups to histone 4, histone 3, or also histone 3. Yeah. And then there are other possibilities up here, other ways. Histone 3 lysine 4 methyltransferase actually will lead to activation. So pick a particular example. That's what I'm saying, because methylation doesn't just do one thing. It does different things, and they're very context specific. So when you make your little chart and you're learning these for the test, just pick one. And it'll be one of these things in between the arrows. And then, yeah, write down what it does, inactivating or activating if it's on this side, and you're good. You are good. And you'll need that for arginine methylation as well as lysine methylation. And since all we know that arginine methylation does is this sort of activation, you can just say that histone 3 or histone 4 arginine methylation, and that's going to be good enough. We won't need a specific number because there's not as much subtlety with arginine methylation, at least yet, in our current understanding. So, we talked about lysine methylation. Let's talk about arginine methylation. And arginine methylation is, of course, almost as complex, just not quite as complex as lysine methylation. So with arginine methylation, you can put one methyl group on there, and that's great. Yeah. Or, and we're going to keep the charge, remember, the entire time, because adding methyl groups does not change the charge, just because of the way that they are added. You can end up with two methyl groups on one nitrogen, so that's an asymmetric dimethyl arginine over here. Or you can have two methyl groups on the two different nitrogens, and that's going to be asymmetric dimethyl arginine. I mean, it's not totally symmetric. You got a double bond and a positive charge over here, but you get it, that the methyl groups are one on each nitrogen. That's what they're referring to with that symmetric or asymmetric. All right. Now, what about phosphorylation? Why not? Let's talk phosphorylation. So, H2A.X, so this is the X chromosome-derived histone 2A variant, which is, when we phosphorylate that, that is going to signal that we have a DNA break in the area. And it can also drive that cell toward apoptosis if that DNA break is not repaired quickly. So as part of the repair process, we're going to come in and we're going to take this phosphate off of the serine on H2A. But yeah, if it is not repaired in time, then this is going to lead the cell to apoptosis, which is programmed cell death. It's basically cell suicide. Yeah. There are other places where phosphates are going to have different effects. So phosphorylating histone 2B is going to lead toward apoptosis, and we don't need a particular amino acid here. Just know that histone 2B phosphorylation, that's going to be enough. It's only really the lysines where we have to specify which one we're talking about, because depending on whether you methylate a certain lysine or another lysine, you can have different effects on transcription, on expression. Yeah. And then histone 3, you can lead to either transcription or to mitosis. Yeah. So that's going to be something that's involved in a cell cycle, basically, overcoming a cell cycle block. All right. So that was histone phosphorylation. And now let's talk about small protein addition to histones. And so there are a bunch of these things. Yes, we're only going to focus on histone ubiquitination. But know that there are other things that you can use to modify histone, such as sumo protein can be added. And sumo protein looks like it's kind of a stretched out version of a ubiquitin protein. Very similar. They appear to be related in evolutionary terms. Like one's kind of a copy of the other that's got a little mutated over time, but still does a similar job. All right, so there are several places where you can ubiquitinate. And let's see, we've got so ubiquitinating histone 2A at lysine 119 can lead to repression of a particular gene. The polycomb phenotype is one that I believe you don't see in humans. I believe this is from Drosophila or something like that. Yeah. Let's see. Histone 2B, lysine 123 can lead to activation and elongation. And other modifications on histone 3 and 4 can lead to DNA repair. So pick one of these off the list and yeah, just tell me what the modification is and what it does. And if you want to pick histone 3 or 4 ubiquitination leading to DNA repair, that's fine. You won't have to remember a particular amino acid because so far we don't know of any differences, any particular subtleties there. Yeah. So let's see. Deubiquitination will have sometimes the opposite effect. So deubiquitination of the ubiquitin ligase addition to histone 2A will have the opposite function. It's going to counteract that polycomb phenotype. And yeah, modification on histone 2B, which the ubiquitin binding protein number 8 binds to, is going to lead to transcription elongation. I don't care which one you pick. Just pick one of these things, learn it, and you will have one piece of the puzzle that you can carry with you and use on the test. So yeah, this is definitely a lecture where you're going to have to do a little bit of work to assemble for yourself a nice little study guide with all the information that we're looking for. So we need to know what amino acid is getting modified. We need to know what histone it's on. We need to know if there's a difference. If it's one of the methyl groups, we need to know which amino acid are we talking about here that is getting modified. But that's only for the lysines, and that's only for lysine methylation that we need the actual position. Then we need to know what the effect is. We need to know what enzyme writes the modification. We need to know what kind of enzyme would take away the modification. So yeah, all of that information is what you are going to put together from this lecture. So it's just going through this. And yeah, if you don't already have good notes on this, take some notes. Make that little chart up, and that will come in very useful on the final and studying for it. All right, now, of course, it's always more complicated than that because why wouldn't it be, right? So you've got all these different histone modifications. Here we're showing histone 2A, 2B, 3, and 4. And modification of one part of one histone will lead to other modifications on other parts of other histones sometimes. So for example, adding a ubiquitin at lysine 23 of the H2B is going to lead to methylation at lysine 79 of histone number 3, and it's going to lead to methylation of lysine 4. But if you methylate that arginine 2 first, then that's going to stop you from being able to methylate lysine 4. So it's this big series. It's kind of like a little computer in that when one thing happens, it has these little ripple effects, and it's going to change other bits in the system, basically. So that you're going to end up making one change that has several other changes, and unless, this is a problem that people ran into when they were first studying modifications. If you're only looking at one change, you can sometimes end up not understanding the process because one change can very much lead to another or can stop another one from being made. And so making a certain change at a certain time can end up having much bigger effects than it otherwise should have. And the way that this happens is that some of the, for example, methylases, methyltransferases rather, and demethylases can work together. So, for example, and they'll be a certain distance apart, and there'll be a protein complex here. And so you'll have methyltransferase over here that's adding methyl groups onto here, but it comes as part of a unit with this demethylase that reaches over, and it's in the perfect spot to demethylate lysine 27 while you are methylating lysine 4 over here, just like that. And so they can work on adjacent parts, and so that's how you can even have modifications on one histone that lead to modifications on another histone that is nearby. And so this is how you end up chaining these effects together. When you modify one, you're going to modify not only other parts of the same histone complex, those other 8 proteins that are in there. Remember, it's 2 copies each of 4 proteins. You're also going to modify some of the histones that are nearby. And so you're going to open up or close down whole stretches of the chromatin at any one time. And there, is what we know. So is there histone code? The answer is yes. It's just that we don't understand it quite as well as we probably should. And so if you were to rank these things from totally shut to totally open, which is red, you can see that you have a lot of different effects. And depending on exactly what is being modified, so here, histone 3, lysine 4 with 3 methyl groups, for example, versus histone 3, lysine 4 with 2 methyl groups, you are going to have different accessibility to different parts of the chromatin. Yeah, ranging from very tightly bundled to very loosely bundled and highly accessible. And so the color code over here is going to show enrichment in expression, like how much you're expressing. So a very dark blue thing will be underexpressed. A very bright red thing will be overexpressed, like twice as much as normal. And so there you can see histone 3 lysine 4 modification. When that is high, that is going to be lead to very open. That's the opposite. Well, anyway, it's complicated. It's very complicated. I think that's the takeaway. I'm not expecting you to memorize anything from this particular chart. And honestly, we'd have to go back into Karchenko et al. Nature 2011 to pick through the details of this, I think. I should have copied out the figured legend on there. So, there we go. Histone code. All we're going to be testing on as we go back through this is you're going to make that table. And so that's some of the information on the table right there. Those are all the different modifications that we're going to be covering. Forget about sumoylation. Forget about ADP ribosylation. You're going to also need to know the effect that they have. So there are certain effects, and like effects of a particular acetylation, an effect of a particular lysine methylation, a particular arginine methylation, and here's the nice chart for all the lysine and arginine methylations, so you can just pick one off of each of those. A particular kind of phosphorylation, and I'd say go for the H2A.X phosphorylation. That's a nice easy one to remember, and that's going to be involved in DNA break repair. And then for ubiquitination, I would say why not go for DNA repair once again, any modification, ubiquitination of histone 3 or histone 4, which is going to happen on a lysine. It hasn't said which lysine, but it just means there's a couple of them there. Those are going to be the results of that ubiquitination. Yeah, put that together in a little table, and you've got yourself a good study guide. So get out there and make yourself a study guide, study up for this, and thanks very much. I hope you found something interesting in this. I certainly think this is a very interesting area of biology.